So here we've got the second lecture in the series. Now we're going to go into population genetics. So in the first lecture, we talked primarily about historical perspective of biology, historical perspective of evolution. And now we're going to get into a more modern perspective. We're going to look at really what's called Hardy-Weinberg equilibrium and a lot of statistics and a lot of math. Now, it's not going to be scary math. I'm going to hopefully walk you through it step by step so you understand everything that's going on. But what we're going to find out is natural selection is important, but it is not by any far stretch the end-all be-all of evolutionary theory. There are 4 other components that we need to deal with before evolution can occur. Heck, we don't even know the definition of evolution yet. We've been talking about descent with modification. So that's what we're getting into today, what we modern scientists consider evolution to be. And I will say you will never hear scientists arguing about whether evolution happens. Evolution happens. Though what you hear an argument about is the definition of evolution. So to begin, we need to talk about populations. A population is a group of interbreeding organisms that are going to be at the same place at the same time. Because really, it's super hard to breed with somebody if you're not in the same place with them at the same time. It's not like a mail order thing. It might actually for humans now, it kind of is sometimes. But in general, be together. Salmon swimming upstream. If you've never seen this, it's insane to watch. But you know, the streams become, depending on the color of the salmon, they can become very pink, and all you see are these writhing bodies going upstream. Bears will hang out by the edge and just pick off salmon, eat whatever they feel like, pick off another one. It's really easy to do. It smells terrible because there are bears killing fish, and then it's not like the bears clean up after themselves. And then they have feces everywhere, but the forest grows really well. I learned something super cool when I was up in Alaska this summer. Was it this summer? Yeah, it was this summer. And I learned about salmon because I'm the type of guy who goes on a cruise and I'm like, let's do all the science things. So I learned about salmon. Do you know there are 5 types of salmon? No, because all we think is there's one type. And it turns out the Alaskans only export the low-quality salmon. So check this out. I can do this. So there's 5 types of salmon. There's the chum. And I remember that because it's like your thumb, right? Chum. And then you've got your pink eye. Yeah, which are, oh, no, I'm sorry. Then you got your pink, which are pinky, right? Then you've got, so pink, chum, and then you've got your king salmon, which is the biggest one. You've got your chum, your pink, your king, your sockeye. So it's like your pointer finger because that's how you sock somebody in the eye. And then, oh, there's one more. And it's because of the wedding ring. But anyways, 5 types of salmon. They export the chum and the pinks because they're the cheapest and they don't taste as good. Kind of cool. That's what I learned. I've also learned that much like Michigan is a hand, Alaska is the hand like this. Things to know, because when you're pointing out where things are, I'm in Juneau, which is like, let's see. Juneau is right here. Thank you. Wow, well done. Yeah, well, that's just the coho. Well done, well done. Which one tasted best, King? Say it again. Yes. They're full of energy, and they're delicious. Here's the deal. It's all about sex, guys. Life is about sex. Surprise, I know, you're college students. It's not like you didn't already know that. You can define a population. Oh, wait, I forgot. There are not many, like, high school, homeschool kids in here, right? Sorry, sometimes. I got them in my 101 class, and I've got to remember what I'm talking about in there. Okay, good. Life's all about sex, guys. You can define populations by who you can breed with. If you can breed with them, they're in your population. If you can't, not your population. Humans have almost a global population now, but even now you still have to, people are like, you can breed with anybody around the world. It's like, yeah, but not really. It's not like there's not much migration between here and Siberia. A little, I'll give you that. But it's not like you're hopping on a plane to go find a date for next weekend. You could, because you're giving up here. But it's possible. Usually, a population is a small, confined group. Now, when I was teaching in Danville, that was really easy to discuss with them, because people dated within that very small 10-mile radius. In fact, there are people that would never leave that 10 to 30-mile radius around the city in their whole lives. It was nuts. I was like, have you guys been to the beach? And they're like, what's a beach? No, they weren't like that. They were more like, no, we haven't, because we have no money. And I was like, sorry. You guys, though, are totally, I'm sure you've left this Midlothian area at some point to go somewhere, right? Yes, right? Did you meet somebody while you were there? No, because you don't like to talk to people. No, because we're all that way, right? I don't like people. But you could have. You could have. So a group of horses, a herd of horses, is an interbreeding population. That group of horses is not going to breed with another group of horses on a different continent. Populations. They're defined by who you can mate with at a given point. Over time, populations change. They're dynamic. They're constantly changing. Here's an example. This is the first Congress. What do you notice about the picture of the first Congress? What? They're all wearing wigs. They're short. They're a bunch of old white guys. I mean, let's just be honest. That's the first thing you notice. It's just all totally pale. You look at the current Congress, and okay, truthfully, still mainly a bunch of old white guys, but you can occasionally see a woman in there. There are some different colored skin tones. Not many. I love how, though, you can usually find the women because the guys are wearing, like, all dark suits, and then, I don't know if they had a dress code or something. It's like, wear red. You can see a few different colors, but it's changing over time. It's slow change, but it is change over time. This change over time can occur due to what are known as polymorphisms. What does poly mean? Poly means many. So new forms, lots and lots of new forms. The traits that display this variation are called polymorphisms. So here you see sheep. We all agree these are sheep, right? And what color are these sheep? White, except for that one. That's black. Variation. It's a polymorphism. Let's talk about sheep and sheep evolution. Because again, we're back to selective breeding. Really, sheep are something that we bred for. Most wild sheep were not white. The white sheep is the polymorphism. That's the, currently it's the, what we know as the wild type allele, but it used to be the mutant. There weren't many white sheep out there. Why would there not be white sheep? What would being white do to sheep compared to other colors? It makes them stand out. So if you're a white sheep in the middle of a field, you're the one the wolf's going to see. Make sense? So then humans came along and we said, man, we really like eating these sheep. Let's protect them. And then we started saying, let's protect white sheep. Why did we select for white? Why did we want them to breed white? Yes? That's one I hear a lot. Yeah, wool can be dyed easily. Because we were totally, back when we were like, we need to eat food, we're like, but we want to wear the right color. No, wool can be dyed easily. That's definitely one factor, but why else? Same factor as before. So we can see them. Does that make, why would we want to see sheep? So we can know where they are. Shepherds, right? Remember they're the guys with like the sticks that go in big thing? They have a dog next to them. What'd you say? Shepherd's crook, thank you. Because I don't remember words. Shepherd's crook. And you've got these sheep, and they're white, and you got a little dog. He's going to sit at the top of the hill and look down, and he wants to see his sheep. Now, if they're black and brown, they blend in. But if they're white, he can be like, top of the hill, 1, 2, 3, 4, 5, 6, 7, 8, I got all my sheep. Nighttime comes. Time to go in. It's easier to find sheep that are white than they're black. I mean, that's a big reason. That way we keep our food, and because that's what it is, food and wool, where we can see it. So we bred for that. Now, there are other colors now. I mean, there's still other colors because those are the original colors. But polymorphisms happen, and then those variations can be selected for or against. Polymorphisms are, at their heart, caused by variations in alleles. Again, what is an allele? Right, an alternate form of a gene. A difference in the gene. Alleles are also called polymorphic genes because they have many variations. So here you might have one allele that's producing purple flowers. Here you have one allele that would cause white flowers. The most common type of polymorphism is caused by single nucleotide polymorphisms, or SNPs. They are by far the smallest change and the most common. So this SNP is caused by, this is sickle cell anemia. Sickle cell anemia is caused by the replacement of one nucleotide in one amino acid in one gene. And it makes blood go from this shape to that shape, the sickle cell shape. Single nucleotide polymorphisms are caused by a single change in a single nucleotide, right? Single nucleotide. Here we've got GGC is suddenly GAC. One nucleotide is changed. These are the most common polymorphisms and lead to semi-functional or maybe even more functional genes. When you get a variant form of a gene, probably caused by a single nucleotide polymorphism, you end up with alleles. So here we have on one chromosome an allele that has one variant for the gene. Maybe it makes purple flowers. Here we have another variant that makes maybe white flowers. These are two alleles for the same gene. You'll notice they're at the same locus, the same location on the chromosome. And these chromosomes are homologs. They're very similar to each other, but slightly different. Now, this is all stuff you learned about last semester, right? You recall it? Or at least you heard about these things. So now we could, how many chromosomes, how many homologs do we have for each chromosome? That is, let me put it a different way. How many chromosomes do you get from your mom? Oh, yeah, 23. Okay, you're right. How many unique chromosomes do you get from your mom? One. And you get one from your dad. And that comes together and makes two. You go from haploid gametes coming together and fusing to get a diploid zygote. That means that you could possibly have two of the same allele or two different alleles. You could be termed homozygous, having the same zygosity, so the same alleles, or heterozygous. You'd have different alleles. You guys have heard of these two terms, right? Sorry, you're like, yes, please move on. I want to be done with this. So if they've got the same alleles, they are homozygous. If they have different alleles, they're heterozygous. We're going to take those alleles, because we're not dealing with, you guys remember Mendelian genetics, right? You dealt with that, and you're like, I never want to see it ever again. You had to do Punnett squares. I'm like, Mendelian genetics, no, Punnett squares, oh yeah. We're not going to really deal much with Punnett squares. We're done with that, because that's an individual. What we're concerned with is a population. So we're going to take all of the alleles in the population and throw them into a big pool called the gene pool. And then randomly, we'll figure out the chances of randomly drawing out one pair of alleles versus another pair of alleles. So I want to point out real quickly, because it's caused confusion before, these are warthogs. This is not how warthogs reproduce. They don't go to a warthog like gamete pool, dump in all their gametes, and baby warthogs come out. I don't know why that was an issue, but somebody pointed it out and said, well, I saw the slide. This is just a a graphic of what may happen in a gene pool. So we've got all of these possible alleles, big B, little b, big B, each representing a certain variant. Then during meiosis, those are split. So we have individual alleles in the gene pool. So now's where we start getting into math. First thing we're going to figure out is what are known as allelic frequencies. And an allelic frequency is going to equal the alleles of a specific type over the total number of alleles. We'll do some problems. We'll work this out. So allelic frequency is the alleles of a specific type divided by the total number of alleles. So in order to figure out the total number of alleles, you count the number of organisms. You multiply by 2, because each organism has 2 possible alleles. Here we've got a bunch of mice. How many mice are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 mice. Thus, if each mouse has 2 alleles, how many total alleles are there? 30 alleles. All right, going through, we're going to count up the number of white alleles. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Yeah, I got 13. You guys have about the same? I'll just go ahead and write 13 down. I think 13's right. Good? Thanks. So that means we have 13 over 30 white alleles, and we have how many dark alleles? Now, there are 2 ways of doing this, and this is awesome. Check this out. You could count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 over 30. So that is the allelic frequency of the dark alleles. But what's the easier way of doing it? Those of you who understand some math. What? From? That total number, right. So the total number is 30 over 30, which is 1, minus, say, 13 over 30, will give you 17 over 30. All of the allelic frequencies are going to add together and equal 1. So far, so good? All right, I'm going to give you another, before we move on to genotypic frequency, give you another of these allelic frequency ones. If I was to have, I don't know, let's say, give me a total number of organisms. I don't care what number. 45, thanks for being so easy. 45 total organisms. Can I bump it up to 50? Would that work for you? I think that'll make everyone's life easier. We're going to go with 50 total organisms. 50 total. Right? We've got big A, big A, big A, little a, little a, little a. So far, so good? That's the possibilities. How many of these 50 are going to be big A, big A? Any number. 17. How many are going to be big A, little a? 13. Who said that? Thank you. That makes life so much easier. And thus, there are going to be 20 little a, little a. How do I know there are 20? 17 plus 13 plus 20 equals 50. So far, so good? All right, so to determine allelic frequency of big A, how many total alleles do we have? We have 50 organisms, so how many total alleles? Each organism is diploid. 100. All right, so for big A, how many big A's are there? How many big A's are here? 17. Okay, so there's 17 of that big A, and how many of that big A? 17, so we have 17 plus 17 plus, how many big A's are here? 13. So we end up with, is that 47? Is that what you guys get? 17 plus 17 plus 13? 47 over 100. So what then is the allelic frequency of little a? You can do it. 53 over 100. Make sense? Any questions? Because I'm going to have you do another one. Because that's the way I'm rolling today. And then I have to talk less. All right, so let's go with 50 individuals. I don't know, 10, 10, 30. So 10 are big A, 30 are big A, little a, and 10 are little a, little a. See if you can figure that out on your own. So the answer is 50 over 100 for both, or half and half, or 0.5, however you want to do it. In this course, for me, a number is a number is a number. You can write it as a decimal, you can write it as a fraction, you don't have to reduce it. You're good. I can interpret as long as you put a number in there. So we've got, for big A, 10, oh, that's a nice marker, 10 plus 10 plus 30, it's the little things in life, over 50, I'm sorry, over 100. So it's going to equal, big A is 50 over 100, or 0.5, and then 10 plus 10 plus 30 over 100 equals 0.5. Questions on allelic frequencies? Now remember, allelic frequencies all have to equal 1. Next up, we've got genotypic frequencies. Here we're looking at the genotypes. 3 genotypes. We're going to take the number of individuals with a specific genotype over the total number of individuals. Using the same data as before. Big A is 10, big A little a is 30, little a is 10. How many of them have big A? Not trying to trick you. How many are big A? 10. And what's the total number? 50. So our genotypic frequency for the homozygous dominant is 1/5, or 0.2. Big A, little a. How many big A, little a individuals do we have? Not trying to trick you. 30. Total number of individuals is 50, and thus you have 3/5, or 0.6. And little a is 10 over 50, which is equal to 1/5, or 0.2. What happens if you add up all of the different genotypes? 1. So far, so good? Any questions on that? The big difference between this and allelic frequencies is you're no longer multiplying by 2 because you're looking at individuals and not at alleles. Now we've been using big A, little a, big B, big T, little t, all these random letters. In population genetics, it's a lot more codified. There are a lot more rules to it. Now I'm not going to kill you on any of the rules, but I'm going to teach it the way it's properly done. In general, P equals the dominant allele. Q equals the recessive, or the frequency of the dominant and the frequency of the recessive. The reason we use P and Q is because you can't confuse a P and a Q. They're just sort of reverses of each other. So in this case, big A is going to be, if you look at the fish, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 A's over a total of 20 alleles, because there are 10 fish. So 10 times 2. And that's going to give you 0.7 equals P. Typically, these are almost arbitrary. As far as Q goes, it's going to be 6 over 20, which equals 0.3. Again, what do you notice here? 0.7 plus 0.3 is what? 1. When in doubt, it's all going to be set equal to 1. This is a key equation. People ask me all the time, and I want to let you know straight off the bat, can I write down these equations on a note card and bring them to lecture or bring them to an exam? No, you cannot, because you're a college student. And here's what the equation is. P plus Q equals 1. Any question on that? I mean, we've derived it several times. The frequency of the dominant allele plus the frequency of the recessive allele must equal 1, because 1 is the whole population. Questions? Okay, now here's where we play the tricky game. So, if P plus Q equals 1 for a population, and you're looking at the way organisms breed, if I was to take P plus Q and multiply it by P plus Q, we do the FOIL method. Do you remember that from algebra? So we get P times P is what? P squared plus PQ plus PQ, so inner, outer, outer than inner, plus the last ones, plus Q squared equals 1. And thus, P squared plus 2PQ plus Q squared equals 1. And that's called Hardy-Weinberg equilibrium. I love these names. Godfrey Hardy and Wilhelm Weinberg. If there were two, like J.K. Rowling should have written those names. Those are straight up Harry Potter names right there. Wilhelm Weinberg. Who comes up with that? So Godfrey Hardy, Wilhelm Weinberg. The story goes, I want to say, so one is a mathematician, and one's a statistician, and one was a biologist. And I want to say that Hardy was the statistician, and Weinberg was the biologist. And they met at a convention where they were at a bar. And Weinberg was describing what goes on in biology. And Hardy, who'd never heard of this theory before, said, hold on, we can write an equation for that. They literally wrote it on the back of a cocktail napkin and then published it after that. And that's how great science happens. Not necessarily drunk, but at least a little bit in a good frame of mind. What this means, I mean, it's really, really easy to just say, you know, memorize this equation and run it out. But I want to tell you about what all of this means. Because hopefully understanding the practical application will make it make a little bit more sense. P squared is the frequency of the homozygote, because you look just the homozygous dominant. So P squared equals the frequency of the homozygous dominant allele. 2PQ is the frequency of the heterozygote. And then Q squared is the frequency of the homozygous recessive. So we have two big formulas we need to know. P plus Q equals 1, and P squared plus 2PQ plus Q equals 1. And then if you understand, P squared is the frequency of the homozygous dominant, 2PQ is the frequency of the heterozygous, Q squared is the frequency of the homozygous recessive, and P is the frequency of the dominant allele, Q is the frequency of the recessive allele. You should be fine. In fact, I'll finish writing more of that. So P is the frequency of the dominant allele. Q is the frequency of the recessive allele. So let me ask you a question. See if you can solve this. P plus Q equals 1, right? So if I have P, we'll make it easy. P is 0.5. Q is thus what? Because P plus Q equals 1, so 0.5. In this population, where P is 0.5 and Q is 0.5, how many homozygous dominant individuals would you expect? What frequency of homozygous dominant would you expect? 25%, 50%. What's the answer? 25? How would you get it? You're right. I mean,
you just do 0.5 squared. So.
Right. P squared, frequency of the homozygous dominant. P is 0.5, so 0.5 squared is 0.25. Because you have to move the decimal. Don't forget to do that. Okay? What if I was to give you P of 0.1, Q of 0.9? What's the frequency of the homozygous recessive? Did somebody say it and I just missed it? What? It's not 9 out of 10. This is the frequency of the, so 0.9 is the frequency of the allele. But I want to know the frequency of the homozygous recessive. Yes, you've got it, Rachel. Tell me. 0.18, yeah, or 0.81. Yes, yeah, no, it's okay. It happens. You're right. I saw where you're going with it, though. 0.81, 0.9 times 0.9 is 0.81. How about this? Same data, the frequency of the heterozygote. What? So you're using this data. You want frequency of the heterozygote. It's okay. I'm going to call you anyway, Rachel, so it works out. No, you don't have to think much. Okay, I'll call him Courtney. It's okay. Whatever. Whatever 2 times, what? 0.9 times 0.1 is? 0.9 times 0.1 times 2. Yes, that's correct. So what's the answer? So what she did was 2 times P, 0.1, times Q, which is 0.9. Oh, come on. Are you using a calculator? What's? 0.18. Yeah, 0.18. Okay, so let's talk basic math, guys. 1 times 9. It's still 9. Times 2. All right, now we've got 2 spaces behind the decimal, so we shift the decimal 2 places, so it's 0.18. Okay? Yay. All right. The reason this is so cool, in my mind, because now we're looking at graphs. The reason this is so cool that under certain circumstances, the allelic frequency will never, ever change from one generation to the next. You may have a difference in the number of one genotype versus another, but as little a decreases, the frequency of big A, little a, and big A begin to increase. And it all balances out. It all always equals 1. So you get these cool graphs. But that's only under certain circumstances. So some math problems for you guys. And these are going to be like the problems we do in lab. This is where I wanted to get us so that we completely understand what's going on in the next lab. Within a population of butterflies, or moths, or whatever you want to call them, the color brown is dominant over the color white, little b. 36% of all butterflies are right. So only knowing how many are purely right, white, 36%, and that is the recessive, I need you to tell me the percent of the population that are heterozygotes and the percent that are homozygous dominant. Can you work that out? Go ahead and give it a shot, and then we'll do it on the board. You will, by the way, have several questions like this on your test. So the key to this problem is 36% of all the butterflies are white. If 36% are white, so 0.36 are white, what is that going to be equal to? That's the frequency of what? The recessive, the homozygous recessive. So that's Q squared. So if the frequency of Q squared is 0.36, what does Q have to equal? 0.18? It's actually not 0.18. How do you get the, how can you go from, it's 0.6 because you have to take the square root of 0.36, which is equal to 0.6. Now, if Q equals 0.6, what does P have to equal? 0.4. And if that's the case, the frequency of the heterozygote is 2 times P times Q, otherwise known as 2 times 0.6 times 0.4, which is equal to 0.48. Is that right? I think so. The rest of you are like, yeah, it looks right to me. I think that's right. So 0.6 times 0.4 is 0.24 times 2 is 0.48. And P squared is 0.4 squared, which is 0.16. So 0.48 plus 0.16 plus 0.36 all equal 1. You want to try another one about moths? Just see if you got it. What? You can't or you can? Oh, good. That's seems to me this is really simple. That's why I'm trying to break it down as far as I can, but sometimes I get lost in it. Try another one where this time we're living in a place where we've got a whole bunch of white trees around, and 81% are white. So now we have 81% of all the butterflies are white. So I want you to tell me how many are homozygous dominant and how many are heterozygous. All right, so we got 81% are white, which means that Q squared equals 0.81. So Q has to equal what? 0.9, because it's the square root of 0.81. Therefore, P equals 0.1. So 2PQ equals 2. Hey, Courtney. So 2PQ here, what's that going to equal? 0.18. Good job. Did you use your calculator on it? Good job. I'm so proud of you. And then therefore P squared is going to equal 0.1 times 0.1, which is 0.01. And 0.01 plus 0.18 plus 0.81 equals 1. Little bit harder problem. Talking about human blood typing now. You guys remember human blood typing, ABO blood typing? It's a codominant trait, so you can see both. You're either blood type A, and if you're blood type A, you have A allele and an A allele. If you're blood type B, you have a B allele and a B allele. If you're blood type AB, you have an A allele and a B allele. It's really easy so you can tell things. So in this population, there is nobody with type O blood. They got killed off in some sort of disaster. It was a horrible biological issue. Everyone with type O blood's dead. So all that's left, conveniently enough for us, are 200 people, 75 of which have type AB blood. 200 have type A blood, 75 AB, and 25 have type B blood. So we're a population of 300. Total population, 300. You know exactly what the blood types are. I need you to figure out the frequency of P and Q, or the frequency of A and B. Okay? That's your goal. Figure out the frequency of A and B. Remember, this is not doing exactly what you just did. Hardy-Weinberg's not involved right now. You're just figuring out the frequencies of A and B. Give it a shot. So how many total individuals are there? 300. So how many then, if there are 300 individuals, how many alleles? 600. So we know our bottom number is 600. How many big A's are there here? 400, because it's 200 plus 200. And how many big A's are here? 75. So we have 475 over 600. And then for this one, we could do it two ways. We could figure out 75 plus 25 plus 25, which is 125 over 600. Or we could have taken 475 over 1 minus 475 over 600. Either way. Any questions on that? Yes? Not at all. Yeah, yeah. Don't worry, we're going to even get into like fitness values and everything. You've got to figure out what to use in what situation. If I'm asking for allelic frequencies and I give you get lots of data like this, it's probably just going to be added up. Hardy-Weinberg is going to be really useful, especially if you are just given a recessive phenotype. So let's do it. Okay, I'm not going to tell you which one this is. You have PTC testing. Hey, in Bio 101, did you guys lick paper ever? No, you never did a PTC test? Okay, PTC testing is awesome. I have to find some PTC paper and have you, it's fun. There is a chemical in Brussels sprouts and in broccoli called phenylthiocarbamide, PTC. And it is very, very bitter. But here's the deal. Only some people in the population can taste it. If you have a dominant allele, you can taste the bitter taste of broccoli and Brussels sprouts. So we'll go with, I don't know, big T. You can taste it. Little t, you don't have the proteins you need in order to taste broccoli, the bitter taste of broccoli and Brussels sprouts. So you think it tastes fine. So then when people are like, how do you not like Brussels sprouts? You're like, how do you like it? You literally cannot, you're biologically cannot tell what they taste. It's really interesting. So little t is a non-taster. So what I used to do is I would take PTC paper and go around the room. And you put it on your tongue. And people who could not taste were like, okay, it's paper. Why'd you have me put this in my mouth? But the reaction of the people who could take it was phenomenal. Because it was like, bleh, and they'd have to run and leave the room and get a drink. Now, it works for me because I know I'm not a taster. So I'd be like, yeah, I'll try it. Tastes like paper. But going around the room, it was fun to watch the reactions. But apparently we don't do that here. Because I'm big into torturing my students, and apparently nobody else is. We have 215 people. We have 215 total people. Of them, 150 can detect the taste. So we'll call them 150 are tasters. Now I'll take you to the next step before I let you loose on this. This is where people oftentimes make a mistake. If 150 people are tasters, how many are non-tasters? Total of 215, 65. No. Yes? Yes. 65 are non-tasters. All right. So I want to know what is the frequency of p squared, 2pq, p, and q. Can you figure it out from that data? You do not need to simplify things or turn them into fractions if you don't want to. All right, so what we'll end up doing is saying, all right, we've got 65 non-tasters out of a total of 215. So what is that going to make q squared equal to? Q Q squared is the frequency of the homozygous recessive, right? Looks like none of you went this route. Frequency of the homozygous recessive then was going to be 65 over 215. Do you see what I did there? Does it make any sense whatsoever? Yes or no? Okay. No. Okay. So look at it this way. We're looking for the frequency of the homozygous recessive. Okay. The non-tasters are homozygous recessive. That's the only thing you can ever know. You don't know whether they're homozygous dominant or heterozygous. You can't tell a difference by looking at them. I mean, they still freak out when they eat broccoli. But it's those non-tasters. They're the ones who you absolutely know what their alleles have to be. Lower taste. They have to be recessive. So if Q squared is 65 over 215, what does Q equal? And this is what's going to drive you guys nuts and you're going to hate me. Q then equals the square root of 65 over 215. And you could just leave it like that. Thus, P has to equal what? P plus Q equals 1. That's 1 minus the square root of 65 over 215. Therefore, P squared equals 1 minus the square root of 65 over 215 squared. And 2PQ equals 2 times 1 minus the square root of 65 over 215 times the square root of 65 over 215. And if you leave it like that, I can read it just fine. If you want to go through and do all the decimals in the calculator and all that, you can do that too. I'll let you use a calculator on this. I'll tell you, I'm not going to do that to you on any of the tests. I'm going to make them nice and simple round numbers. Do you have questions on this? All right, I'm going to do one, maybe two more. See what this one is. Oh, albino squirrels. Albino squirrels freak me out. I saw one and I was like, no! And they're like devil squirrels. Albinism, this is, by the way, more like what will be exactly on your test because it's a very simple one. You should not need your calculator for it. You can use it. I will mock you, but you shouldn't need it. Albinism is a recessive trait. If the population has a 4% rate of albinism and the population is in Hardy-Weinberg equilibrium, what would be the expected genotypic ratios of the homozygous dominant and the heterozygote? Well, and then I put what would be the expected phenotypic ratio? Phenotypes, what they look like. Well, if 4% are white, it makes 96% brown or gray, whatever. So go ahead and figure out the genotypic ratios. You got this. We've got a population with 4% albinism rate, which means what does 0.04 equal? Q squared. And if 0.04 equals Q squared, what does Q equal? 0.2, because 0.2 is the square root of 0.04. And if Q is 0.2, what's P equal? 0.8. So if that's the case, what is P squared, or the homozygous dominant? 0.64. And then the heterozygote's 2PQ, so 0.2 times 0.8 times 2 is 0.32. And then we have 0.64 plus 0.32 plus 0.4 equals 1. Any concerns? A little bit harder. It's not bad, but it's a possible scenario that you need to prepare for. After graduation, you and 49 of your closest friends, as if you had 49 friends. So you, let's go with 25 males, 25 females total. Total population of 50. So 50 total individuals. decide to go on a plane, and the plane crashes, and you you land on a desert island, and it's really awesome for you. No one finds you because it's deserted island. Nobody knows where it is, and you decide you're going to start a whole new population because you're stuck here, and you're just going to make it work. Now, you are totally isolated. 9 of your friends carry the allele for sickle cell anemia. They don't have sickle cell anemia. So 9 of your friends are going to be big A, little a. The rest of them, totally normal. Big A, big A. So how many are the rest of them? How many? 41. 41 that are totally normal. 9 of them that have this trait, but they're not displaying it, so they're fine. Assuming that the frequency of the allele does not change as the population grows, because now you and your friends are mating, you're having a bunch of kids, life is good on this island. What will be the incidence of sickle cell anemia in the next generation? In the next generation, what will little a be? First thing you need to do is what? Determine your allelic frequencies. So what is the frequency of big A, which will be P? Total number of organisms? How many total are there? 50. So what do we have to do to that? Double it. So we have over 100. Made that nice and easy. How many big A's are there? So 41 plus 41 is 82 plus 9 more. So you end up with 91 out of 100 are big A, which is P. And for Q, therefore you have what? 9 out of 100. Because P plus Q has to equal 1. All right, so now we know P and Q. What's the frequency of the the homozygous recessive going to be? Square it. So Q squared, therefore, has to be 9 over 100 squared, which I think is 0.081. No, one more zero, yeah. 0.0081. Very low incidence of sickle cell anemia. That's that. So the key note to this is don't go on a plane with your friends and crash into an island.
I would hate to be like the master not handle this until that one kid who got anemia. It's like, you're like scientifically impossible.
Statistically, you shouldn't exist. But the deal is, they do. And as we talk more and more about sickle cell anemia itself, sickle cell anemia, the trait for sickle cell provides an advantage on tropical islands. Because if you have sickle cell, if you're a heterozygote, you are immune to malaria. Whereas all of these big A's, they're going to get malaria and die before they have kids. So you get a balancing thing going on. And that's what we're going to start looking at. How these things balance out, how all of these different alleles can exist still and not just be bred out of the population entirely. So we've talked about Hardy-Weinberg equilibrium. We've talked about what happens when allelic frequencies don't change and what you can expect. You can expect every generation, one generation to the next, there will be absolutely no change in allelic frequencies. The problem is allelic frequencies are always changing, all the time. There are forces acting on them. Now, natural selection is one of those forces, Darwinian evolution. People have talked about Darwinian evolution for 100, 200 years now. But we know that there's several other mechanisms involved, and other mechanisms may be just as consequential. You have to have new mutations, new variation. That was one of Darwin's postulates, right? You have to have variation. Well, that variation comes about through mutation of the DNA. You could have one allele providing a reproductive advantage. One allele allows them to survive better. That's straight up natural selection. the population is so small that random chance matings may have an effect on the allelic frequency. So if we have a very, very small population, if one organism was to mate more often than one other just by chance, that will change the allelic frequencies in the next generation. If you have migration, immigration, or emigration, you're bringing in new alleles or you're taking your alleles out of the population, causing those allelic frequencies to change. And then if one gender or the other has a preference for mates, that is, there is non-random mating. If any of these postulates falls through, then Hardy-Weinberg is in disequilibrium and you have evolution occurring. And that's what we're looking for. Evidence of evolution is found in allelic frequencies. We're going to go through each of these. Now, I guarantee you this slide, probably more than any other slide in this entire lecture, is going to be important for your exam. For me, this is huge. This tells you, one, you know Hardy-Weinberg. Hardy-Weinberg's allelic frequencies don't change. Evolution is not occurring. It's what we term our null hypothesis. And these are all of the exceptions. When any of these are happening, Hardy-Weinberg is in disequilibrium and evolution is occurring. So expect an essay. Hardy-Weinberg is a mathematical construct put together by two British men in a bar in the 1940s. It's not real life. You guys know real life is more complicated than what we can predict on a page. And maybe eventually we can figure out a mathematical equation to everything, but we don't have that yet. So these the allelic frequencies are always changing, and a big example people always use is the peacock. The peacock has this giant fantail. It doesn't help it survive. It certainly, if you're you got two peacocks, one with a really, really long tail, one with a really short tail, that really long tail peacock is going to be eaten first by a predator. But the deal is that tail helps it mate, which means that its alleles get passed on before it dies. So you get allelic frequency shifts that you wouldn't otherwise see. So yeah, Charles Darwin was the first one to propose all these selection mechanisms for evolution, but we've come a really long way since then. Over time, the genetic variation in a population shifts. It moves around. The structure of the population changes. This is a map of Europe. To me, this is really fun. Actually, it's not a map of Europe. This is a map of alleles in different countries, a certain set of alleles. These alleles are not really selected for. They just happen to be prevalent in a population. And what they did was they correlated the frequency of the allele to the language that you spoke. And you'll notice that this green blob over here, Spanish, you're getting this group of individuals who speak Spanish, all have roughly the same allele. There's not much other color in there. France, same thing. There's a little bit of that Belgian allele in there, but there's still, in France, you have a distinct set of alleles from Spain. Belgium, a distinct set of alleles from Spain and France. Britain has hardly any other color in there. Germany, you're getting a big mix. And between the Hungarians, the Czechs, the Slovaks, you've got a lot of mixing. Now, why is that? Why is it that certain language groups will all have a certain allele? It's not like your language is passed on through your genes. Why do certain language groups have the same allele throughout their population, and other language groups don't have that allele? Is it accent? Well, it has something to do with their language, yeah. But what is it? Yeah.
They're more likely to mate or like be able to communicate with people who speak the same.
That's the big thing. Lesson number 1, college, right? If you can't talk to somebody, you can't mate with them, or you shouldn't anyway. It's really hard to mate with somebody who speaks a different language. So what you're seeing is the British, who are one, on their own little island, and two, kind of homophobic, not homophobic, what is it? Xenophobic, thank you. They're kind of xenophobic. They don't really want to, they don't get much mixing, much mating. So there we go. Oh, I do have a joke that goes with this, though. So what do you call somebody who speaks 2 languages? Bilingual, right? And if not, you didn't know that English word for the day, bilingual. What if they speak 3 languages? Trilingual. What if they speak 1 language? American. I love that joke. And the sad part is, it's really, really true. Now, you've got one way that you're going to change allelic frequencies is through new mutations. A mutation is an alteration in the genetic sequence. And they happen. They happen through a lot of different mechanisms. One is just simple exposure to sunlight, simple ultraviolet radiation. You get a slight change in the DNA. You could have variation through crossing over. You get changes in the genetic sequence. This variation is able to be acted upon by natural selection. This is the process where you get a heritable beneficial trait to increase in successive generations. I mean, Darwin's finches are the one we really harp on. You start off with a basal form that has an intermediate beak, and as it specializes to its different ecological niche, you get new forms arising that are just beneficial. I like this cartoon. The bear-proof garbage containers have caused a natural selection of higher IQ. The idea is the bears that can break into the garbage containers are the smarter bears, and thus they're able to pass that on to their offspring. And this is one of my all-time favorites, evolution in action, natural selection. A bunch of guys sitting in a pool with stuff plugged in the center, and the power strip is just floating on 2 flip-flops. I was legitimately going to put a new picture up here. I saw a wasp nest this weekend, and it wasn't a little wasp nest. It was like one of those big ones in the trees. And I was like, if I take a picture of me swatting at it, I could put it on this slide. And then my students will learn something. And I was like, no, that's stupid. Yeah, it was really bad. So I didn't do that. I will say, I've seen a lot of natural selection in action, a lot of stupid tricks, stupid college student issues. I used to teach at UNC, and it was snowing. Now, people in North Carolina are usually not used to snow. I'm not sure what it's like around here. Do people like stop driving, everything shuts down, the world's over? Oh, the bread and milk is gone? Okay, never mind then. Same all over then. Well, there I am teaching. There was an ice storm. There was ice all over the sidewalks. And I'm teaching on the first floor lab. It wasn't this concept, but this is a concept very similar to this. And I see a student walking down the stairs. Now, this student, in 27-degree weather, was wearing a short skirt and really high heels, trying to walk down icy stairs. And I stopped the class. I was like, okay, everybody, you have to stop listening to me now and look out the window. So we all turned and looked. And there she is, very unsteadily going down, and she slides the rest of the way. And I was like, and that is evolution in action. Downside is, she's still going to be able to have kids. Adaptation. Natural selection promotes adaptation so that the next generation is better able to survive than the current generation. So here's basically natural selection. We got a bunch of crows, right? These crows prefer green beetles because, I don't know, they're delicious. You start off with a population that is a higher proportion of green and a lower population of brown. Over time, the crows keep eating the greens and the browns begin to rise in numbers. So you have a shift in the frequencies, evolution occurring. Until finally, all the green is gone and you have fixation of the brown beetle. Now, a few things can happen at that brown stage. I mean, this obviously isn't the end. Evolution is an arms race. What would happen if these guys only ate green beetles and now all that's left are brown beetles? That's exactly what happens. Either they eat brown beetles or the crows adapt. Some crows prefer the brown. They start to increase in number and their population evens out. Another thing that could happen is the crows don't adapt. They all die. And then the brown beetles take over the earth. I mean, these are things that could happen. You have to remember that it's a two-way street. As a prey evolves an adaptation, the predators are adapting to that. And I don't know that we cover it, but that's called the Red Queen theory. Have you guys ever read Alice in Wonderland, or at least seen the movie? One of them, I don't care which. There's a scene in Alice in Wonderland where the Red Queen tells her you have to run as fast as you can just to stay in the same spot. That's evolution. We are not jumping forward in leaps and bounds. For every step forward we take, our predators go forward with us. And it just keeps going back and forth. Look at bacteria. Bacteria are very quickly evolving. We think we got them on the ropes. We're like, yeah, we use science. We defeat you, bacteria. And like, oh, no, we're immune to that now. And we're still killing you. We're like, oh, yeah, well, we come up with something new, and it's awesome, and you can't stop it. And they're like, yeah, totally did. And that's the conversation that's happening between us and bacteria. So adaptation is constantly occurring. Increases the ability to reproduce. Now, we can measure that reproductive ability in something called fitness. I will tell you, the hard math is done. Hardy-Weinberg, that's as tough as it gets. But we still need to come up with measures of reproductive success. You've got to choose what it is you're looking at. Now, this is cool because you as the scientist get to choose it. You might choose the number of eggs something lays, the number of possible gametes it produces. That is a measure of fitness. You might choose the number of offspring produced, or the number of offspring that survive to adulthood, the number of offspring that reproduce. You as the scientist are going to be responsible for defining what your measure of fitness is. Now, I told you before, the whole point of life is to reproduce. I mean, people are like, what's the point of life? It is reproduction. By any measure, this family wins. They're like they had a show on TLC. No, not the Brady Bunch. Not the Brady Bunch. The Duggars. Yeah, so these people, their whole, like, they just keep pushing out kids to the point where their kids are taking care of their kids, which is kind of a cool system when I thought about it, because it's like, well, I don't have to do the work and I'm lazy. But having this many offspring is a huge measure of reproductive success. I don't know, how many of you guys have one sibling? You and one other. Okay. You guys, your family compared to this family, you guys go on to reproduce, have a normal size, and by normal I mean 2 kids, right? These people go on to reproduce, and they just start churning them out. Suddenly, you have a whole town full of this, and you have maybe a van load. So they're winning. They have higher reproductive success and are passing on their prolific genes. What about money? Oh, resources. Resources are going to absolutely limit what you're able to do in any environment. You, frogs, birds, doesn't matter. If you don't have the resources to survive, which was, I think, one of Darwin's postulates too, right? There's always going to be more organisms produced than the environment can sustain. I don't know how. I think they're sustained now through like Kickstarter or something. We don't have that option. Well, we have the option, but nobody's going to pay for it. I should start a Kickstarter. They probably did need the show. You'd need a show, or at least people who are really gullible to give you money.
Or
One of those husband things?
but like not,
Like a husband who just pays for everything? Got it, okay. What are those called? Like sugar daddies, right? You didn't want to say sugar daddy in class? That's okay, I said it in class and I'm going to put it on YouTube. So, there we go. So that later my dean has evidence. So we've got to measure these things. We've got to measure fitness. With modern natural selection, you've got a new mutation that creates a new allele. That allele may be better at performing. It may produce a better protein. It may produce an inferior protein. But either way, if that allele impacts the ability of an organism to reproduce, it's impacting their fitness, and it's going to have an impact on the allelic frequency in the next generation. So some alleles are going to enhance fitness. We got 2 I think these are platies, but I'm not sure. Platies and guppies look the same to me because they're fish. Some of them are going to allow you to survive better. These are found in streams and rivers in Hawaii. Now, in Hawaii, there are two types of riverbeds. There are black sand ones, and there are white sand ones. Black sand riverbeds with these fish aren't going to help its survival. Why? Yeah, because it stands out. Birds come along, they eat the fish, that's that. So in black streams, you tend to find darker fish. In light streams, you find lighter fish. And that's because one generation after the next, you're getting adaptation. You had a mutation so that you had this lighter fish. That fish doesn't survive well in the dark, but survives really well in light, and then passes that allele on to the next generation. Individuals with more beneficial alleles are more likely to survive and contribute their genes to the next generation. So it's just a bunch of animals mating. There we go. Whether they're protists fusing, sharks mating, turtles mating, doesn't matter. They're passing their genes on to the next generation. Those that succeed have succeeded for a reason, and those genes may help. I find this hilarious that sharks, when they mate, sidebar, shark mating. You guys ever seen sharks mate? Okay, so what sharks do, the male shark will bite the female flipper, the female lateral fin, bite onto it and hold tight because he's a shark and she's a shark and they're freaking big. And then he plows her head into the ground. Kind of rough. Turtles are weird. If you guys, again, turtles will, they mate, and they can mate for a very, 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 very long time. And it's not just because they're slow. It's that they're turtles. Turtles will mate with anything that is vaguely turtle-like. If they're successful, they pass their genes on. So over generation after generation after generation, the allelic frequency for the beneficial allele is driven upwards, leading to a net increase in the fitness of the populations. So I was saying we have to measure fitness, and it can be measured in lots of different ways. But you need to describe the contribution of one genotype to the genetic composition of the next generation. We can do that by establishing a new mathematical value called W. W is our fitness score. It's given a relative value of 1. I'm running out of space to write. For instance, these are chickens, right? Our measurement is a group of chickens. We're going to measure by the number of eggs they produce. That makes sense, right? You measure, you can figure out how fit a chicken is by counting the eggs it lays. We have genotype AA produces 12 eggs. Big A, little a produces 5 eggs. Little a produces 1 egg. Which of these, I'm not trying to trick you, is the most fit? Yeah, the homozygous dominant, big A. Because of that, we are going to assign it a fitness value of 12 over 12, which is 1. All of the rest are less fit. For this big A, little a, then, we're going to assign it a fitness value of 5 over 12, because 12 is our highest. For little a, it'll be 1 over 12. So if we have a bunch of chickens, and I say, wait, any questions on how I did that? This is way easier than Hardy Weinberg, right? Let's go with, instead of 12 eggs, we'll have big A, big A, big A, little a, little a, little a. Give me a number. I don't care what number. Doesn't matter at all. 10. Okay. Give me another number. 4. Give me another number. 8. Which of these is the most fit? 10. So you're going to make that value equal to 1. 10 over 10. What's big A, little a's fitness value then? 4 over 10. Little a, 8 over 10. And all this does is it tells you who's the most fit in any given generation. You could use anything you want. You can measure it in any different way. Here we've got, let's see, lizards. Lizards with a genotype big A, little a produce an average of 2 offspring each. Lizards with genotype big A, or little a, produce an average of 1 offspring each. What are the relative fitness values? Which one has the highest fitness? The heterozygote. What's its fitness value then? 1. What are the other fitness values? 1/2, because it produces roughly half the offspring of the other. There we go. Fitness. Now, fitness will, how well these offspring are able to produce leads to changes in allelic frequency between one generation and the next. Selection of these fitness values, so selection can be seen in lots of different ways. We might have what's called directional selection. Directional selection is what you think of when you think of typical Darwinian evolution. If you are selecting against one extreme, then you're getting higher values in the next generation. This is a graph. Okay, good. So here's one generation, here's generation 2. Original population, new population. This is selecting against, I don't know, let's call this axis short, and this is the number of individuals. So here you got shorter individuals, there you have taller individuals. You're selecting against the short ones. So what happens in the next generation? You get taller individuals, okay. Basketball players are tall, right? We're good on that? Let's say hypothetically, people prefer to mate with basketball players compared to short people. I lost it there. If they prefer to mate with basketball players, then you assume that, or you believe the next generation will be selecting for higher sizes, taller sizes. What will happen in the generation after that if you continue selecting for height? You get even taller organisms, and taller and taller. This is called directional selection. You're moving in one direction. Stabilizing selection is where you have an intermediate phenotype produces an advantage. In this case, I don't know, you don't want to mate with short people still, but tall people keep hitting their head into the doorway and dying. So you've got two selective events. Short's not getting any mating, tall's not getting any mating. So all of the individuals are getting into be this intermediate size. You're selecting against the extremes. This works out actually a lot in fish. You guys have been to the fish store, right? Like a PetSmart or something. And you've seen these brightly colored fish. Brightly colored fish, like brightly colored red fish, are on one end, brightly colored blue fish on the other end, and right in the middle is sort of this dull gray color. Nobody wants to buy dull gray ones. But in the wild, bright colored blue, bright colored red gets eaten. It's only that intermediate phenotype that survives. So that stabilizes the population. That being said, you might have diversifying selection. With diversifying selection, you're selecting for two extreme ends and against the middle. We just gave the talk about the fish. Brightly colored red fish, brightly colored blue fish, gray fish. If you are selectively breeding, you don't want gray fish, you want either blue fish or brown fish. So you get rid of the, you don't let the gray ones mate, and you only let the brightly colored ones mate, and it leads to two distinct forms. Back to our height thing with people. If short people just keep mating with short people and tall people keep mating with tall people, you're not going to get that intermediate phenotype. You're going to have a population beginning to form, divergence. This is one way of getting speciation, which we'll talk about later. Here we have moths, butterflies. In the center, you have the sort of intermediate phenotype. The intermediate phenotype looks delicious, not to us, but to birds and things. The one end of the spectrum, though, looks like an unpalatable species. It looks poisonous. The other end of the spectrum also looks poisonous. So birds are not eating this, Birds are not eating that. Birds are only eating in the middle. So you're getting an increase of alleles in the next generation of the two extremes, and you're against the center. So that's diversifying. One of my favorites and most interesting is balancing selection. There are times when a heterozygote serves to maintain a genetic polymorphism. And this can be found in the major histocompatibility complex of humans. Let me walk you through this. Anybody had anatomy before? Okay. Did you learn about the immune system and how the immune system works? Did you talk about the MHC, the major histocompatibility complex? No? You just learned about like how blood cells do this, that, and the other. So this is the underlying mechanism behind how we're able to recognize diseases. Your body produces these templates, these little proteins that can recognize intruders. Those proteins are going to be encoded for by the major histocompatibility complex. Now, if you have all of the exact same allele in the MHC, then you're not going to be able to produce nearly as many proteins to detect disease. Did you follow me on that? It's advantageous to be heterozygous at the major histocompatibility complex, which means if you have different alleles there, you can produce lots and lots of different proteins to recognize lots and lots of different disease agents. It turns out, after some studies, women can detect by scent whether or not a man has different alleles at their MHC than they do. That is so weird to me. Apparently, we can still detect things by scent. The way they ran the experiment, and I kid you not, is they had a bunch of men and a bunch of women work out without any deodorant on. Now, I know that right there is signaling, I don't want this. Then they took their shirts and hung them on a line, and they had the men and the women go through and smell the shirts. Significantly, the women were able to determine whether or not men had different alleles at their major histocompatibility complex because they preferred that scent to any of the other scents. Men could not tell in the slightest, but females can. What this does, the more heterozygotes for balancing selection, the more heterozygotes you have, the higher your fitness. They were smelling like the shirts. And it's just based on what was secreted from sweat. Isn't that weird? But you have to admit, there are certain people that you're like, okay, you smell better than that person. Yeah, you know what I'm talking about? And you know, if you've ever had a breakup, that you've got that weird smell memory that you got. You're like, yep, smells like them. And then you have the feelings. Not good with feelings. Balancing selection. Following up with that, a better example would be sickle cell anemia. Sickle cell anemia is bad, right? You get sickle cell anemia, your blood cells are in the wrong shape, they clog up your veins, you die. If you are a homozygote recessive. There's no fitness advantage to having sickle cell anemia. Literally, your fitness is seriously reduced because you die oftentimes in childhood.
Doesn't it stop you from getting malaria, though?
But wait, there's more. Yes. Sickle cell anemia absolutely prevents you from getting malaria. But it doesn't help because you're dead. You know, you die of your blood disease. Now, the thing is, though, like you were bringing up with malaria, in sub-Saharan Africa, if you're homozygous dominant, that is, you don't have any sickle cell at all, you're perfectly normal, you get malaria, you die in childbirth, or not childbirth, childhood. So there, your fitness value is low. It's the intermediate, the heterozygote, that's totally immune to malaria and doesn't have to worry about sickle cell. So that heterozygote is strongly selected for, while the two other ends, the homozygous dominant and homozygous recessive, are selected against. And that leads to a higher prevalence of sickle cell anemia in populations who have been living in sub-Saharan Africa and to an extent now Latin America. Yep. Absolutely. 25% of the offspring likely to have sickle cell anemia, 25% likely to be totally normal, get malaria and die, 50% of them totally immune. So you're able to increase the incidence of that heterozygote, keeping both the dominant and the recessive alleles still in the population. They don't go extinct. They're not that strongly selected against. And then we got sexual selection. With sexual selection, one gender or the other chooses their mate based on specific sexual characteristics. And this is hilarious to me. Whenever you see something odd in the animal kingdom, some weird trait, some crazy thing that you're like, why does it have that? Likely, it's for sex. It's not like it's some sexually selected trait. Let's talk about the narwhal. You guys have seen, you know what a narwhal is? Ever heard of a narwhal? Narwhal is sort of think a dolphin with a giant horn sticking out of its mouth. It's what? It's an elf? Oh, it's in elf. I thought you said it was an elf, and I was like, it's not an elf. It's a narwhal. It's a but you know, whatever you want to go with, I'll go with it. Yes, it was in Elf. I remember that. It popped up. Yeah, good movie. One of my favorite Christmas ones. Right. Why does it have this big long tooth? Well, I gave you the answer. It's for sex. But why else would it have it? Because you know there are people out there who don't think it's for sex. They think it's for something else. What would this big giant tooth sticking out of its mouth be good for? Fighting. Right, males fight each other, right? Why are they doing that? Impress the ladies. My sister got into an argument with me. My sister's, she's trying to be a lawyer. So my mom's a lawyer, my older sister's a lawyer, and my younger sister is really trying hard. And I got to support her in this. So she wanted to argue with me about the narwhal. And it follows up on a moose argument that we had years earlier. But this is my family, right? She said, we were talking about the narwhal, and I was like, yeah, the narwhal uses, Thanksgiving conversation, the narwhal uses its tooth in order to choose mates. And she's like, no, it uses it to hunt. And that made me think, what does it do? She said, well, it goes and it stabs fish with it. And then I felt really bad for the narwhal. So I thought, if it goes and stabs a fish with it, how's it going to get it off? And then I imagined, then back to the moose conversation, my younger sister also swore that moose eat squirrels. And she said, I said, well, why do you think that? She said, well, that's why they have horns. They just chase the squirrels up the tree. And they stab them with their horns. It's like, no. It's like, well, why else would they have horns? I was like, well, horns are something that look weird, right? It's all about impressing ladies. They lock horns. But she said, no, it's to chase squirrels. And then all I could think of was the image of this moose with dead squirrel corpses on its horns, trying to get the squirrels off to eat them by scraping them against a tree. But there we go. So don't think everything's for gathering food. Things are usually, weird things are for sex. Intrasexual selection. Two members of the same sex compete in order for the opportunity to mate. This is why moose have horns. This is why deer have horns. Deer horns are not strong enough really to withstand a major predator. It's not like they're going to stab a wolf with it and then be fine. No, they push each other around. Elephant seals slam up against each other. And these things weigh like 1.5 tons each. They do some major damage with their blubber. Kangaroos, they do this whole fighting thing. But they can be dangerous. You know, kangaroos, Australia, right? We know Australia is not the best place. Kangaroos fighting each other will gore and eviscerate each other because they have these claws on the bottom of their feet. So they jump back, lean back on their tails, and they rake their claws across each other's stomachs. If that happened to a human, we would be eviscerated. Your intestines would spill out. There's all sorts of intrasexual selection. These are usually males competing against other males. It's kind of weird. It's kind of fun to watch. You know, you see these males fighting it out, and you look and you think, yeah, that happens in humans. And you guys have seen intersexual selection in humans. You've seen males, especially male humans, do this all the time. You guys went to high school, right? Right? Yeah. It happens there like all the time. Think about it. Just about every single fight in a cafeteria, what happens? You've seen fights. Again, this region has fights in cafeterias, I assume? Okay. What happens? What's the first thing that happens? What? No? Nobody watches. Oh, come on, guys. You know the sequence of a fight in a cafeteria. No, I'm not even saying what I'm saying. What do they do? That's not the first thing, though. They don't just sit down and start hitting each other. They start yelling and screaming, but before they yell and scream, they stand up, don't they? Absolutely. And usually to the effort of knocking over a chair. Boom, I stand up, and then they puff themselves up. Like their chest cavity expands dramatically. And they go right up to each other, don't they? Half the time, it's not just yelling at each other from like across the table like this, like politicians do. No. They come up, and they're butting chests up against each other like, I'm bigger than you. Look how big I am. They're literally doing exactly what elephant seals do. They're like, boom, boom. And then when they're doing that, they're like pushing each other back, or it becomes a contest of wills. Push each other forward, push each other back. Who's going to give up space? Who's going to give ground? Just like these guys do. And then, yeah, sure, they start hitting each other. But they don't hit each other to kill each other. Usually, I don't think when I moved here, I didn't see many fights resulting in death. Usually, they fight until they figure out who's dominant. One of them slinks away. One of them is down. The other one wins. Does that make sense? They win, and what do they do when they win? They stay puffed up. This is my territory. Behold all who see me. I am the biggest. High school boys are just like intrasexual animals in selection. Then we can get to intersexual selection. This is where the female actually chooses the male. Displays. These are the big colors that you're going to see. The anole lizard will puff up its throat sac, gets very, very large, and you can see it turns bright, bright red. Now, that's not good for it. Bright red in a lizard will attract a bird. Usually it's camouflaged, but when it's trying to mate, it's like, hey, check it out. I'm dangerous. I have big red thing. Not only does it do that, it starts calling out to females. It starts croaking really loudly, saying, hey, I'm over here. I'm over here. Predators also follow that. Red-breasted robin. Female robins, they don't have red breasts like that. No. Because they're, I'm not going to say smart, they got the good genes. They've got the genes that say, I'm not going to die from predation. The males, the more red you have, the more likely you are to mate. The also more likely you are to be eaten by a predator. There's a balancing act there. And we talked about the peacock and the peahen. This is what the peahen looks like. Not nearly as impressive as the peacock. But she's got those the traits that will allow her to survive predation. The male, though, really focused on those traits that will allow him to reproduce. If he tried to escape a wolf, the wolf could grab the long tail, pull him back out of whatever bush he was hiding in, and eat him. And once they get, yes?
So lifespans are really different.
Lifespans among organisms are very, very different. Not just, lifespans are about the same if the male can survive. It's all about predation. But the big thing is, how can I put this? There are different forces at work choosing alleles in females than there are in males. So, yeah, the male might not live as long. In fact, females live longer than humans too, by like a decade at least. My wife's already planning what's going to happen when I'm gone. Apparently, she's going to open up some sort of youth center / bookstore. I was like, have fun. She's like, why can't you support my plans? I'm like, I'm going to be dead. Do whatever. I don't care. Once again, with intersexual selection, we can see connections to high school boys. and actually college guys too, let's be honest. Displaying. Look at me, I'm big and I'm strong. And the only way the reason I say this is I've driven behind people and their cars are so loud. You know the ones I'm talking about, right? Where it's just not just loud music, it's the bass. And that bass is like shuttering windows. The deeper the bass, the more likely that an old lizard is to mate. And I'm thinking that there's some guys out there who feel like the deeper the bass, the more likely it is for them to mate. I don't know. Oh, people revving their engines. Why do that? It's so, yeah, I know. And you're a girl who would choose who to mate with, right? You're not going to be like, that guy has a louder engine than that guy. I should mate with that one. No. Yeah, no, it makes no sense. Yet people do it. It's all about showing off. It's all about saying, look at me, look how big I am, look how many resources I have. So in case I haven't really emphasized it enough, these sexual derived traits are going to be balanced by predation. A characteristic that's appealing to a mate, long tail, bright colors, is also going to make it easier for them to be eaten. So while one allele is being acted for, it's being selected for this sexual trait, other alleles, other forces are acting against it. So you get a balance. That's why you don't have peacocks with like 30-foot tails. There comes a point where it's too much. Another major concept is genetic drift. Genetic drift is going to be changes in allelic frequencies due to absolutely random chance. These occur most often in very small populations. So let me give a quick example. Let's see. Let's say, so crow, wake up. Crow, hey crow. Crow, okay. So you got a, sorry.
Sorry, it's the cold air. It's messing with my head.
It's cool. You've got a blue jacket. You're the only person in here wearing blue. Good on that? Okay, that wasn't the whole thing. Don't worry. I just needed your blue jacket. You can go back to sleep as soon as this is done. You got a blue jacket on. Nobody else has a blue jacket. If that fan above Crow was to break and fall and slaughter him, he is dead. He cannot reproduce now, right? He cannot pass his inherited blue jacket on to the next generation. Will there be blue jackets in the future? No. It's gone due to random chance. Now, in a larger population where there were more blue jackets and this fan came and crushed Crow, it would be fine because there are more blue jackets elsewhere. Does that make sense?
And now I'm dead.
Now you're dead. You can go back to sleep. If you look, here we've got a population that's very small, and here's a population that's very large. These alleles are not being acted on by selection. They're neutral. There's no selection involved. It's all due to random chance. And you notice that in a smaller population over time, one allele or the other will go to fixation. That is, it will hit a frequency of 1 or 0. Once an allele hits a frequency of 1 or 0, it's either all the way through the population or completely gone. It will stay that way until there's a mutation or a migration event. So these are going to really impact small populations much, much more than large populations. Which again, I believe is exactly what this is showing. Here you see what happens with 25 stoneflies in an experiment, and here's 500 stoneflies. They're looking at two alleles that are not acted on by any sort of selection. Very quickly, the smaller population goes to fixation at either a 1 frequency for the allele or a 0 frequency. But with a larger population, you see it sort of hovers around the center. Random chance. One type of genetic drift is called the population bottleneck. In a population bottleneck, there's the large population is very suddenly decreased in size. So it's sort of like our example with Crow, except instead of Crow dying because he's under that, everybody else dies and it's only Crow and one other female left. All then that would be able to pass on to the next generation are lots and lots and lots of blue jackets. All the other alleles are gone. This happens a lot. This happens a lot in nature. Happens a lot because of humans too. We hunted whales. Sidebar, humans are super big into oil, right? That's like our thing. We're like, we find oil, we get it, and we run with it. Well, at some point, we figured out there's oil in whales. And we said, we want that oil. We will hunt the whales. So for 2 or 3 generations, actually several hundred generations, we hunted whales until the whale population was diminished entirely. We're down to just a few hundred whales. When you lost all of those other whales, you've lost all of their genetic diversity. All that's left is a little bit of, a few alleles left. Then the population rebounds. We have a lot of regulations now protecting whales, and there are a lot more out there. A type of population bottleneck is called a founder event. Here, instead of decreasing the population naturally rebounds on its own, you're going to have a small population moving to a new geographic location and starting off that new population with only the alleles it brought with it. One really cool example of that is the island of Krakatoa. Krakatoa exploded. I think it was late 1800s. Krakatoa used to be an island, and then it exploded. It was the loudest sound ever recorded in the world. It was heard all the way around, all the way on the other side of the world when this island went boom. When they went back, after it exploded, they sent ships back to it, the island was gone. It was a volcano that had built up so much pressure, it just completely blew itself up. I assume it was sort of like that scene in Moana. But the idea is everything was wiped out over time. The next 15 years, lava kept spewing up from the remains of the volcano underwater, and you got a whole new island formed. That new island had no life on it. No, literally no bacteria, no fungus, no plants, nothing. It was all just lava rock. So now we have this whole new place where there's no life. Over time, life started to come back. First that came back were the seabirds, because really there's nothing living there, and it's safe. So they landed there, they dropped their feces, and it started to produce a soil. Next, coming in on the wind and waves were grasses. And that makes sense, right? You get seeds from plants. You get plants growing around this island. This is a bird paradise. There are birds everywhere, trees starting to grow, grasses. And then one day, there was a large storm. And a Komodo dragon washed up on the shore on a piece of driftwood is what they think. It came from a neighboring island. And it just happened this was a female Komodo dragon that was pregnant. So when she came to this island filled with birds, her favorite food, she started a nest, she laid her eggs, and she ate. And then her children hatched, and they ate. And her children then grew up, and they wanted to mate. Well, who can they mate with? Just each other. So the only alleles on this population for the Komodo dragon were what those offspring had. So whatever this female and her mate had, those are the only possible alleles around. These Komodo dragons are so genetically similar to each other simply because of an extreme population bottleneck, a founder event. The same thing happened in Scotland. Scotland is well known for red-headed people, right? The reason for that is during a migration event, red hair is not common. As people were migrating from the British Isles to Scotland, it happened that a small percentage of them had this very rare red hair. Well, they mated together. Their descendants, went on to mate. And as they did that, there was a higher and higher incidence of this red hair allele, or many red-headed alleles. So in an original population, we have just maybe 1 out of 100 people. If the 5 or 10 people that went to the new population, one of them happened to have that red-headed allele, suddenly that frequency shoots way up. And then The future populations are based on that founder population. A consequence of this is lack of genetic diversity. You end up with clones everywhere. These organisms have the same genes. That's where we get lab rats. All lab rats look the same because they're pretty much genetic clones of each other. In order to get lab rats, what they did was they took a rat and another rat, and they put them in a cage, and then rat babies came. Then they took those babies and crossed them to each other. And they took those babies and crossed them to each other, generation after generation. Why in a lab would you want to have genetically identical offspring? It's a control, right? You're going to want to make sure that whatever drug you're testing is not having some effect based entirely on one organism's genotype. You want to make sure you can test and retest and test again to see if there's, to make sure the effect is not caused just by the genes, but is caused by the actual drug. Yes? Yep. No, it wouldn't. Can you give me, probably, before we get into incest, can we, no, don't laugh, because we're going to talk about, give me like 3 slides. Okay. No, today's concept, incest. We're going to talk about it a bit. I got in trouble for making it, so I'm going to put it up here, and we're going to learn it. But before we get there, I do want to talk about the neutral theory of evolution. We've talked about the theory of natural selection. There is a lot of variation. Most of the genetic variation between us is caused by neutral genetic changes, those changes that do not impact your fitness at all. You have these little mutations, one generation to the next, little mutations that distinguish you from your siblings that has absolutely no impact on fitness. I mean, well, you could think of several probably. What are some traits that you don't care about in a person when you're looking to mate with them? Let me ask you some questions. As you go on a date, do you ask somebody, so what's your blood type? It's not something you care about? No. Maybe it is, I don't know. Maybe you're like, I just need to make sure so I can get a transfusion. Do you ask somebody, can you taste phenothiocarbamide? What do you think of broccoli? I mean, these are not major things you're going to ask on a date. Those are going to be things that don't have an impact on selection. This neutral theory is also known as non-Darwinian evolution, or survival of the luckiest. It's pretty much, it all runs through genetic drift. A lot of the mutations, a lot of the changes that happen within your proteins within your DNA happen in the 3rd codon. Okay, we're back to transcription, translation, replication. Remember, when a ribosome is translating a piece of RNA, it takes 3 nucleotides, and it's going to look at those nucleotides and say, all right, these 3 nucleotides equal this particular amino acid. Do you recall this? Yes? No? And then the rest of you aren't responding, so you don't count. That 3rd nucleotide has what's called a wobble. It doesn't matter too terribly much. Look at the genetic code. 1st nucleotide, 2nd nucleotide, 3rd nucleotide. So U and C matter a lot. U, C. But UCAG in the 3rd base doesn't matter at all. You're going to get serine no matter what. Here, threonine, you have AC, UCA, or G. That 3rd codon only matters a little bit in a few of them, not many. So there's not as much selective pressure there. You have a different allele, but it's not represented in what actually gets translated. So even if you and somebody else have slightly different alleles, you may produce the exact same protein, so it doesn't have any impact on your fitness. So that's neutral theory. You could have random chance, random small changes. Migration, Anytime you migrate out of a population, you are going and taking your alleles with you. It's like when you leave a game and you take your ball with you. You're like, I'm going and I'm taking my alleles. You'll suffer for it. Or you could come into a new population. You're like, I'm coming in, I'm carrying all my alleles with me. Check it out. Now we're going to be more fit. Migration is a way of introducing new alleles to a population and changing allelic frequencies. So this could be the result of immigration, alleles coming into the population, or emigration, alleles leaving the population. In general, immigration is better than emigration for a population. You want as much variation as possible within a population. Migration between two populations ends up decreasing the differences in allelic frequency between those populations. So if we, let's say we've got us in here, and then we've got a whole bunch of red-headed people over in the anatomy class next door. If we had migration, we'd have immigration. We would have some red-headed people coming in here and bringing their alleles, so the next generation, because apparently we're stuck in here and all having offspring, the next generation would have more redheads in this class, but less over there. If some of you guys migrated over there to mate, then you're taking your alleles with you. Eventually, over time, there would be free migration and uniting the populations. But if it's just, it all depends on the amount of genes moving. Then we got non-random mating. Some organisms prefer to mate with other organisms that display a certain trait. This would be either homospecific or heterospecific crosses. Homospecific means the same cross. You prefer to mate with things that are similar to you. Heterospecific is you prefer to mate with things that look different than you. There might be some mechanical problems with mating. Certain dog breeds don't breed well together because they just don't fit. I mean, the small male would need like a step stool. It's not going to work. So yeah. So we've got our homospecific crosses, the like ones mating, and when the like ones mate, what do you expect is going to happen with their offspring? When likes mate, they're going to produce, okay, these two birds mate, and they produce what color bird? Blue. These green ones mate, what do they produce? Green, yeah. Pink ones, what are they going to produce? Pink ones. Typically, when you cross to something that looks like you, you're going to produce offspring that look like that. If you were to take two purebred beagles and breed them together, what are the offspring going to look like? Purebred beagles. Questions on that? That's called a homospecific cross, the same. Heterospecific crosses are going to result in increased heterozygosity. Here's where you get your mutts. You take two dogs that don't look anything alike, you cross them together, and you get all sorts of stuff. If any of you guys have ever had, or you haven't had, but if your dog has had a litter of puppies with something that you didn't know what it's from, you look at the puppies and you're like, I don't know. There's a lot of differences in the offspring. And this, Riley, is where we get to inbreeding. Homospecific crosses lead to increased chances of homozygosity, increased chances of the same allele multiple generation after the next. This inbreeding happens when genetically related individuals mate with each other. Inbreeding increases the frequency of homozygosity. So here we've got a snake that has a mutation. It's a recessive allele. Maybe it's albinism. It's not showing it. It's just got one chromosome that's screwed up. It mates with another snake, and they produce a bunch of snakes because snakes make snakes. That's the way it works. You'll notice that you got two that are fully dominant and two that are heterozygous. Now, in a normal population, if it was to outbreed, it would breed more than likely to another snake that was fully normal because that mutation is rare. But in a small population where the siblings might mate together, now suddenly you have an increased chance a 1 in 4 chance of getting a fully recessive individual. You've increased the chances for homozygosity. Inbreeding depression is what people often talk about when they worry about inbreeding effects. You probably have heard all sorts of stories about what would happen if you mated with your cousin. In general, if a homozygote has a lower fitness than the heterozygote, you have what's called inbreeding depression. That doesn't happen all the time. A lot of times heterozygotes are more fit, which is why with purebred dogs, the homozygous purebred dogs tend to have a lot of these medical conditions, hip dysplasia, things like that. Heterozygotes don't really have those. In humans, which is probably what you're most concerned about, there's a lot of intermarriage between cousins and close relatives. It was fairly common in human history right up to the recent past, especially in dynasties, especially among kings and queens. So this, by the way, is Charles Darwin. This is his wife, also his first cousin. It was common. Heck, think about the Egyptians. The Egyptians took it to the extreme. The pharaoh was a god, and thus could not mate with anybody else but another god. Who are the only other gods around? Their sibling. So they end up having a lot of intermarriages within a single family. Early human inbreeding, and you can imagine it happened a lot because there are small clusters of humans as tribes early on. Early human inbreeding, when a group first starts to inbreed, will lead to a lot of what are termed lethal alleles. In your body, you have these ticking time bombs. They're not going to kill you. But if you mate with somebody who is very closely related to you, it is likely they have the same allele, the same lethal allele. 2 copies of a lethal allele will wipe out the offspring. So if you mate with your sibling, I don't recommend it. You will end up with an offspring that may have lethal alleles in two recessive lethal alleles that will kill them. That's what people are generally thinking when they think, don't mate with a close relative because you'll end up with these alleles that will decrease the fitness in the population. Now, that's a deterrent for 1 generation. Given 3 generations, lethal alleles are wiped out. Why? Because the carriers are dead. Once you've mated with your sibling, you had kids, those siblings mate with each other, they have kids, those siblings mate with each other, they have kids. The ones that survive don't have the lethal alleles anymore. That is a stable population. So after several successful generations of inbreeding, most of those lethal alleles are gone, leading to normal fitness in the offspring. And this is how societal inbreeding continues. As long as you continue to mate within a small group that does not share the same lethal alleles, it's fine. Maybe not morally or whatever you want to call it, but biologically it works. It's how you can have lab rats reproducing all the time with their siblings. It's how you can have dogs reproducing with their siblings. The lethal alleles go away. For societies, that's formed a way of passing wealth down from one generation to the next. You guys don't have to worry about that, so it's kind of nice. Again, I'm in a much larger area now than where I used to live. I used to live in, again, Danville, Virginia, where it was totally legitimately a worry for people. Is that person a cousin I don't know about? Should I not date them? And you live there, you know people who've had to have that thought. I'll tell you a fun inbreeding story. It didn't actually happen. So I was in Portugal, and I was just starting to meet people because I was visiting family there. And I met a girl, and I was so excited because she spoke English, and I wasn't that good at speaking Portuguese at the time. And we hit it off really well, and we said, hey, we'll get together later. And I was so excited because I was like 16, and I talked to a girl. And then later on that night, I saw her again, and it turns out she's my cousin. And then I was like, huh. Well, it's the way it rolls. Luckily, nothing happened. Now you guys are looking at me like, this is an inbreeding story, right? Well, it didn't actually happen because there's society things. She was the one that shot. Thanks. I appreciate that. I was just like, well, you know what? I'm overseas. Whatever. No. Yeah, no, we looked at each other and like, oh, it's good to see you. Nice to meet you. Inbreeding can be a major issue in populations that need to be managed. There are certain populations that are going extinct. The ibex, for instance, was going extinct. It was a very, very, very large goat. In order to save the ibex, scientists have tried to introduce closely related species in order to save some of the genes. For the ibex, there was only one male left. That was it in the whole world, one male ibex. It lived in a preserve, and so what they tried to do is get it to mate with as many goats as possible to produce goat-ibex hybrids. Now at that point, they're thinking, well, now we've got the genes saved, but is that goat-ibex hybrid an ibex or a goat? That's where we're getting speciation and definitions. What do you guys think? Would it be a goat? Would it be a goat? Would it be an ibex? It's a new thing? Well, so get this. What then if they turned around and took those baby goat-ibex hybrids and crossed them together? And now you've got something that's 75% ibex. Is it an ibex, or is it a goat, or is it something new? It's an ibex, because now you got more. And then, so this is what they've been doing. Then they took those baby goats and crossed them together. Now you got something that's 96% ibex. It's an ibex, or is it? Think about us. Any of you guys done that, like what's it, 23andMe or some sort of chromosome history thing? If you've done that, likely what you found is there's some Neanderthal DNA in you. That's a totally different species. Does that mean you're not human? Sorry. That's the way it is. Oh, as far as what happened to the ibex, there was one male ibex left in the world. On this managed thing, people took care of it. It died. The way it died, this was a act of God, the species was not meant to exist thing. It was standing under a tree. A bolt of lightning struck the tree, ripped the limb off the tree. It fell on the ibex. The ibex died. Not kidding. True story. Last of the ibex died by death by tree. And we saw it. We're like, we will protect you, ibex. And then the ibex died because of a tree. Are they mean? I've never met one. They're big. They used to use them as meat. Allelic musical chairs. So usually what I do is I'll pull out some alleles. We'll play musical chairs, randomly mate. But I think you guys understand the point. There are 5 big aspects to exceptions to Hardy-Weinberg. New mutations, immigration, emigration, or migration. So migration, mutations, natural selection, non-random mating, and genetic drift, or effectively random selection. There we go. So what we've been talking about is what's called microevolution. Microevolution is something that we can experiment with, something we can see in a lab. The change in the gene pool from one generation to the next, to the next, to the next. It is absolutely happening. We can see this with evidence. We can see this with experiments. We can measure the rate of evolution in an organism from microevolution. Macroevolution is much, much harder to look at in a laboratory. Microevolution is what happens from one generation to the next. Macroevolution is how we got to where we are now. It's going to be, well, with our current state of technology, we cannot obviously go back in time and see how things happened. We can't test to see how one species becomes another. So macroevolution is the application of microevolution over a long period. We know microevolution's happening, and if it happens in this way continually, you end up with macroevolution. But you cannot directly test macroevolution. So I want to make that clear, because people are always like, you can't test evolution. You can test evolution as microevolution. You cannot test evolution at our current level with macroevolution. Macroevolution is just sort of the application of microevolution principles on a larger scale. For that microevolution to happen, you have to have new genetic variation. Could be mutation, could be migration, could be anything, but you have to have new variation. And you have to have one mechanism that affects the allelic frequencies. Again, we're just looking at Darwin's postulates. You've got selection, you've got drift, you have something acting on the frequencies, and you've got something creating new alleles. But once we get above that species level, once we're looking at accumulated changes, it gets a lot more theoretical. Do you have any questions? Don't forget, as far as studying for the exams and things, really look at these review questions. They will guide you on what I think is important. Things like, what's the neutral theory and why is it called non-Darwinian? It's not because it's not Darwin. Everybody good? Awesome.